Welcome to your first lecture in Pathophysiology. This course will bring what you learned in Anatomy and Physiology 1 and 2 into application as we look at what can go wrong in the human body. This week we will start the cellular level and see cellular pathology and how human genetics affects diseases and conditions. First, we will review the organelles of the cell and show some of the most common cell pathology and then go to genetics and genetic diseases. Do you recall the organelles of the generic cell as pictured here? Most cells do not have organelles like this. It would be like having a car with all the features at once. Instead, this cell is designed to give you a picture to understand all the parts of the cell. Let's go through the most important ones. The plasma membrane is a phospholipid bilayer that is semi-permeable, which means it lets some substances in and keeps some from going out. It has receptors on the surface and embedded proteins as well. Think of it being like an ocean that is more like a waterbed you can walk on with islands floating in it. How does the plasma membrane move substances in and out of the cell? As you remember, there is passive and active transports that enable the plasma membrane to transfer substances in and out of the cell. Passive goes from high to low concentration and does not use ATP, while active goes the opposite way from low to high concentration and must use ATP. Diffusion is the most common way substances pass through the plasma membrane, and then osmosis is the next most common. Osmosis, as you recall, is the diffusion of water only. It is water traveling from high to low concentration, but it can be stated in another way. Water will travel to the highest level of solute in order to dissolve the solute and increase the water amount. Basically, water wants to even things out. When you place a cell in distilled water that is a hypotonic solution to that cell, then water rushes into it. When you place a cell in normal saline that is isotonic to the cell, and the cell is calm and happy. Therefore, we use normal saline as IV fluid. Finally, a cell in a hypertonic solution, like this one in 3% sodium chloride, will shrivel up as the water leaves it to dilute the liquid around it. It is important to know osmosis in this course as it comes up again and again. As for active transport, the most important one to know besides endocytosis and exocytosis is the sodium-potassium pump. It causes 3 sodium ions to leave the cell and 2 potassium ions to enter, and ATP is always required. We will see this pump again and again in this course. The plasma membrane can be injured, and this will destroy the cell, as what should not enter does, and what should not leave the cell then does so. Free radical damage is the usual cause of plasma membrane destruction. Free radicals are oxidizers. They carry an unpaired set of electrons that will disrupt organic molecules like the plasma membrane. They are known as superoxides, and to remember that free radicals are bad news, recall that oxidation is destructive. If we lived in a world of 100% oxygen, then we would not be here. We would have burned up long ago. Most of our world's gas is nitrogen, and although we need oxygen, too much of a good thing can be bad for you. Antioxidants are the cure for free radicals, so it's imperative that you take these in every day to help your body. Next, recall the nucleus is the brain of the cell and houses our DNA. It also contains the nucleolus that makes ribosomes for the cell. Nuclear pores are found in the nuclear envelope, so ribosomes and RNA can escape. DNA is usually in the form of chromatin while in the nucleus. This allows it to be used for transcription. What is the difference between DNA and RNA? Well, DNA is a double helix while RNA is single-stranded. DNA codes for your genes while RNA is a small piece of those genes and usually codes for one specific protein. DNA stays in the nucleus except at the time of meiosis and mitosis, while RNA readily leaves the nucleus in the forms of rRNA, mRNA, and tRNA. DNA has the base thymine, while RNA has uracil instead. RNA is made from DNA, and DNA makes itself. DNA replication is semi-conservative, meaning that one original strand pairs with a new strand each time to help reduce mistakes during replication. Deoxyribonucleic acid is DNA. The sugar backbone of the molecule is strong and nearly unbreakable, while the weaker bonds of the base pairs can be broken regularly for purposes of transcription and replication. Without DNA, you would not be you. DNA comes in many forms based on how it is folded and weaved. Its most complex form is the chromosome, which only forms during mitosis and meiosis. Most of the time, DNA stays in the form of chromatin. DNA controls all aspects of the cell as it codes for all your genes. Genes are simply proteins that are made from your DNA. Most of the DNA does not get used, but a good portion does code for proteins used in your cells. You get one set of genes from your mother and the other from your father, and between these two genes, you will have specific and unique characteristics that make you the person that you are now. We can measure an individual's karyotype if we can get a sample of their cells during mitosis. This will tell us how many chromosomes they have and what they look like. 46 is the number humans should have, but some have more or less based on the genetic diseases we will look at the end of the chapter. For example, an extra chromosome 21 can cause a trisomy of 21 and result in the very common genetic disorder called Down syndrome. When it comes to the last pair of chromosomes, females have XX and males should have an XY, but we will see later that this too can go wrong. In order for genes to be expressed, they must be made into proteins. The processes of transcription and translation are essential for the genes and DNA to eventually become a protein used in the cells. We will look at this again here in a few minutes, but the process goes: messenger RNA is made from DNA, and that is transcription. The messenger RNA binds with a ribosome and various tRNAs to build the protein in question. That's translation. Remember, the architect or boss of the entire process is the DNA. To better understand genetics, we need the vocabulary. An allele is a gene that is inherited from one parent. Genotype is your genetic makeup, and phenotype is how those genes are expressed physically. Alleles for a gene may be homozygous or the same, or they may be heterozygous or different. Depending on whether that gene is dominant or recessive will determine if it is expressed in a heterozygous state. For a recessive gene to be expressed, it must be unopposed in a homozygous state, while dominant genes will be expressed whether homozygous or heterozygous. One can also be a carrier for a gene with a hidden recessive gene that can pass along to new offspring. There lies the chance that the recessive trait will finally be expressed. Now, most of these dominant and recessive traits are just variations and not harmful, but sometimes they are genetic diseases. Here's an example of a benign set of traits: brown eyes versus blue eyes. If the dark blue box representing the dominant trait is brown eyes, and the light blue box representing the recessive trait is blue eyes, then in the heterozygous state, where there is one parent gives us brown eyes and the other blue eyes, then the offspring will have brown eyes due to that trait being dominant. In the homozygous form, where both are brown eyes, there is no question that brown eye trait will yet again be the phenotype as it is dominant. Now, with both parents giving the recessive trait of blue eyes in a homozygous set of alleles, then the offspring will have blue eyes. This is how genetics works, except there are many more genes which can affect many traits and several that affect the same ones. You can predict what the phenotype of potential offspring will look like based on the genotypes of the parents by using a Punnett square, and this is based on Mendelian genetics. Putting father at the top and mother at the side, you can figure out what the types of offspring will be and whether a recessive trait may be expressed. Rigger Mendel was a monk who studied the genetics of pea plants for many years and is considered the father of genetics. Here's an example of non-Mendelian genetics, as it deals with the last set of chromosomes, or the sex chromosomes. Many traits will fall on the X chromosome, and since boys only get one of these, they will have only those traits, whether dominant or recessive. This is how hemophilia spreads from one generation to the next. Hemophilia is a rare bleeding disorder that affects primarily males for this reason. Mitochondria are probably independent microorganisms that were taken in by eukaryotic cells long, long ago. Mitochondria have their own DNA. This DNA is maternal DNA, and maternity can be traced through the mitochondrial DNA. Mitochondria make ATP for the cell and do so best with aerobic respiration, or the use of oxygen. They are not as effective at this in anaerobic respiration, or the lactic acid cycle. When you run a while, your legs may begin to cramp as the lactic acid builds up from decreasing amounts of oxygen, and the change over to the lactic acid cycle, resulting in lactic acid buildup in the muscles. The number of mitochondria present in the cell depends on the energy requirements of the cell. For example, heart muscle has many mitochondria, as does skeletal muscle. Mitochondrial DNA comes from the ova and therefore is inherited maternally. There are a number of mitochondrial DNA diseases, but there are genetic treatments. Ribosomes are made in the nucleolus of the nucleus and then can be found free-floating in the cytosol or attached to rough endoplasmic reticulum. Ribosomes make proteins. They are made of rRNA, which hold the messenger RNA and allows the tRNA to bring the amino acids over to match the triplet codons on the messenger RNA. This entire process needs ATP and therefore can end if there is a lack of ATP due to hypoxia. Think of transcription like changing audio English dictation into a typed English form. You're still the same language, but just a different form of English. So in terms of transcription for DNA, it's making RNA from DNA. You're still in the same nucleotide language, just a different form. Translation is going from one language to another. Like when the Rosetta Stone was discovered, it allowed for the translation of Egyptian hieroglyphics. In translation, we are going from a nucleotide language to an amino acid language with the creation of proteins from messenger RNA. It is important for you to remember the differences between transcription and translation. DNA never leaves the nucleus, as shown here during these processes. Messenger RNA is made from DNA and then leaves the nucleus and binds to a ribosome, which is rRNA. tRNA then brings amino acids to the ribosome, and they are paired with triplet codons on the messenger RNA, and the amino acid is then joined to others in the chain until the protein is made. There is a stop and start codon on the messenger RNA, which signifies the beginning and the end of this process. Defects in protein synthesis can lead to the death of the cell, and anything that interferes in this process can do the same. Accumulations of fats can also disrupt cellular functions and prevent such imperative processes like protein synthesis. Damage to the DNA will also prevent proteins from being made, as DNA is the blueprint for the entire operation. See this patient here with xanthomas and xanthelasmas from fat accumulation. This is a patient with a genetic familial hyperlipidemia. Endoplasmic reticulum can be rough or smooth. Rough ER makes proteins as it contains ribosomes, while smooth ER makes fats and some carbohydrates. The ER is a collection of membranes throughout the cell, as seen here in relation to the nucleus for illustration. The Golgi apparatus or complex is found at the end of rough ER, as it is used to package proteins. For example, this is done with many hormones like insulin for release from beta cells. Vesicles, much like the plasma membrane, can bind to it. Think of the Golgi like FedEx. Lysosomes are like catfish of the cell, or garbage men. They roam around and destroy waste and invaders to the cell. They are frequently found in liver cells, as these cells are the most common to aid the body in getting rid of toxins and contaminants. Failure of the lysosomes can cause the accumulation of harmful substances. There are so many lysosomal storage diseases, but because we love you, we are only holding you accountable for the top 3. Tay-Sachs disease is the first one. Patients with this disease die very young, usually by age 3, and they have very characteristic cherry red spots on their macula on retinal examination. NIEMANN-PICK disease is next up and also results in early mortality in children. It is mostly brought about by lipid accumulation in the brain, spleen, liver, and lymph nodes. Lastly, Gaucher disease is, and this is the most common of the 3, and is most difficult to identify of the lysosomal storage diseases. Tay-Sachs is the most known for the cherry red spots on the macula of the retina, although you can see it with some of the other ones, but this disease is the most likely if you see it. NIEMANN-PICK has 2 types and results from the lack of breakdown of sphingomyelin from ineffective lysosomes. Seen here are the glucocerebrosides accumulation in the macrophages. These are called Gaucher cells. Stained in pink are characteristics of Gaucher's disease. Peroxisomes are a lot like lysosomes but smaller and use hydrogen peroxide to break down waste substances. An example of their failure is the disease adrenoleukodystrophy, which causes long-chain fatty acids to build up in the nervous system. Now let's learn some pathology terms to help us better understand pathology. Histology is the study of tissues. Biopsy is histological samples taken for analysis. Autopsy is the examination of dead or diseased organisms. Pathognomonic changes are what we look for in order to diagnose known diseases. They are the unique identifying presentations. Atrophy are cells shrinking in size, while hypertrophy is when they grow in size. Here's an example as like a weightlifter lifting weights, you can make your biceps bigger by hypertrophy. Or when the heart experiences increased afterload as an increased peripheral vascular resistance from hypertension, it can become hypertrophied over time from working so hard. If the muscle is not going to be used any longer or is overused, in the case of the heart, the size then shrink and the muscle is wasted or atrophied. Hyperplasia is an increased size of a tissue, but by the number of cells, not an increase in cell size like with hypertrophy. An example is when estrogen stimulates the growth of breast tissue, but maladaptive changes can also result from hyperplasia, and we can see that in keloid formation from scarring. Metaplasia is a change of one cell type to another, and this can be seen in Barrett's esophagus, where cells in the esophagus near the gastroesophageal junction begin to change into more like stomach cells due to constant stomach acid exposure from reflux disease. If left unchecked, this could result in esophageal cancer. Dysplasia is the change of a cell to a deranged or bad cell type. This can lead to cancer and is usually known as precancerous. An example is the dysplasia of cells at the cervix of the uterus in women. These must be treated or they could become cervical cancer. The classic Pap smear is used to check for this. Neoplasia literally means new growth, but in terms of pathology, it could be cancerous or benign. If cancerous, then it is disorganized, uncoordinated, and uncontrolled. There's a lack of cellular contact inhibition. If benign, then it looks organized and ordered. A benign growth, again, is organized, well-differentiated, does not metastasize, and has well-defined borders. A malignant neoplasm is unorganized, poorly differentiated, with poorly defined borders and highly likely to metastasize. Hypoxia is a low oxygenation of the tissues. This can be from hypoxemia or due to other factors like anemia from bleeding. Hypoxemia is the lack of oxygen in the blood. Atherosclerosis is an important pathological concept. It is damage to the arterial walls that can lead to blockages of the arteries. It is initiated by endothelial cell injury. Causes of atherosclerosis are simply what can damage the endothelial lining of the arteries. Hypertension can cause weakening of the walls leading to aneurysm. Diabetes results in glucose damaging the endothelium. Homocysteine directly damages the endothelial lining. Free radicals disrupt the plasma membrane of the endothelial cells. And persistent angiotensin II secretion along with plaque from fat deposition on the endothelium can all lead to atherosclerosis. These are important terms to understand in pathology. Ischemia is when tissue or organs are starved for oxygen for some reason. This can be transient, like a transient event, like a momentary blockage or an anginal with a partial blockage. There is no evidence of cellular or tissue injury here. Oxidative stress is transient ischemia due to disruption of the oxygen circulation, but then it is restored. Infarction is prolonged ischemia that results in injury cellularly and tissue damage, and this is supported with evidence of tissue destruction. For example, when dealing with an MI or a myocardial infarction, the ECG can look the same in ischemia or angina in a myocardial infarction because they're the same electrically. The difference is the positive troponin, which is a muscle enzyme. seen in cardiac muscle exclusively and shows evidence of organ and tissue damage from the prolonged ischemia. Necrosis is the death of tissue or organs from infarction. Gangrene is when the dead tissue or organs begin to decompose and then become infected potentially. If it becomes infected, then Clostridium perfringens is the most common pathogen, and both surgical and antibiotic treatment is necessary to save this area. Apoptosis is programmed cell death. This usually happens when a cell enters G0 on the cell cycle. At this point, if there are genetic mutations or issues like cancer formation, a cell can then undergo this self-destruction without interfering with cells around it. There are no inflammatory markers released, so this dying cell does not cause damage to the cells around it. This method is also used when getting rid of tissues we no longer need, like webbing between fingers during embryonic development and the loss of ovarian function in menopause. This process can be diseased if we lose the ability of apoptosis, resulting in cancers like prostate cancer or breast cancer. It can also cause damage to tissues like the thyroid and hypothyroidism if the thyroid cells undergo apoptosis abnormally. To correct cellular injury, we can do transplantation of tissue or organs, either from oneself, which is an autograft, or from a donor, which is an allograft. Regenerative medicine is done using the stem cells from one's own body. Therapeutic cloning is done using embryonic stem cells and is most controversial. An aneurysm is a weakened arterial wall. This can come from atherosclerosis and from prolonged uncontrolled hypertension. The most common type of brain aneurysm is a berry aneurysm. Named this as it looks like a berry growing when seen on angiogram, either CTA or MRA of the head. Berry aneurysms form in the circle of Willis. Now for some nutritional concepts. Vitamins A, D, E, and K are the fat-soluble vitamins. Protein is essential for nutrition and cellular health, and the lack of protein can lead to kwashiorkor. Marasmus is the lack of all nutrients and generalized malnutrition in children. Now we'll return to genetics and look at genetic diseases. Familial hypercholesterolemia is a very rare and is autosomal dominant, homozygous, and is much more severe than heterozygous forms, which show that this disease has some codominance in its traits. A homozygous child could die from a myocardial infarction, and this makes this disease very important to know. Marfan Syndrome is a rare autosomal dominant genetic disease seen in people with a tall stature that will have back pain, vision problems, easily mobile joints, heart murmurs, and neuropathy in the legs. They could have pneumothorax or an abdominal aortic aneurysm. The best way to remember what these patients look like is to think of Abraham Lincoln, as so many scholars believe that he had this disease. So think of Honest Abe. Familial adenomatous polyposis is an autosomal dominant genetic disease that causes colon cancer, as these patients have numerous colon polyps. Polyps are masses that are on a stalk. Colon polyps can lead to colon cancer, and so these patients must have frequent colonoscopies from a very young age. Neurofibromatosis is an autosomal dominant and comes in two forms. These patients have numerous tumors all over their body, causing disruptions of body systems and functions. A patient with this disease will have cafe au lait spots, which are dark and flat macules, optic nerve tumors, which can cause visual problems, and tumors in the iris of the eye may also be present. Ehlers-Danlos Syndrome is an autosomal dominant collagen disease. These are the circus contortionists. They have hypermobile joints and stretchy skin like this guy here. They can have aneurysm formation and mitral valve prolapse. Cystic fibrosis is a terrible autosomal recessive disease. In the Middle Ages, they said if your baby tastes salty, then it will not live long. This disease causes abnormalities of sodium channels resulting in very salty sweat as well as thick mucus. It's hard for the lungs to get rid of infections as the mucus elevator from the ciliary apparatus is not effective. These patients have recurrent antibiotic-resistant pneumonias, New therapies are allowing patients with this disease to live longer, though. They also must exercise regularly to help keep their lungs clear of mucus. Wilson disease is an autosomal recessive disease causing copper deposition all over the body. Most patients with this disease present later in life with liver issues like jaundice and elevated liver enzymes. It is then found after working up the liver problem. Its defining characteristics include Kaiser-Fleischer rings of the eyes and an elevated serum ceruloplasmin level. G6PD deficiency is an X-linked disease and is the most common enzyme disorder. This disease caused hemolysis of the red blood cells prematurely and can be diagnosed with a CBC and a blood smear. Prevention of hemolysis is key. Prader-Willi Syndrome is a disease causing low IQ, hypothalamic dysfunction, and obesity. One can also see hypotonia, short stature, and hypogonadism. Angelman Syndrome is similar to Prader-Willi Syndrome. It is seen in children and causes more neurological problems. It is maternally transmitted. Huntington's disease or Huntington's chorea is an autosomal dominant disease that comes about in midlife. There is no cure, and the patients know they will have it one day, as others in their family already have it. Some get tested for the genetic defect so they know that they will have it or not, but others elect not to get tested. The disease causes a dance-like movement disorder, which is known as a chorea. Klinefelter syndrome is a disease seen in males with 2 X chromosomes and a Y chromosome. It gives males female-like characteristics like large breasts, wide hips, and smaller testes. Treatment is testosterone therapy at an early age. Turner syndrome is seen in women with only 1 X chromosome and no other sex chromosome. These patients may be spontaneously aborted and never make it to birth, but some do survive. They have short stature, webbing of the neck, and decreased IQ. They lack breasts and have amenorrhea. They need estrogen therapy starting at a young age. Fragile X syndrome is also a disorder of the X chromosome. It results in cognitive impairment and autistic-like behaviors. They typically have large ears and a long face. Down syndrome is the most common mental deficiency and is from an extra chromosome 21 called a trisomy. They will have epicanthic folds around the eyes and a flat facial profile. Figuring out if your baby has one of these diseases can be done with prenatal screening. In fact, it is mandatory given an older mother, as the risk for many of these diseases increase with age. Women over 35, couples who are related, abnormal ultrasounds, women with frequent unexplained miscarriages, or family histories of various genetic diseases can be triggers for those procedures. You can check the blood of the mother, but this is not as accurate as getting blood from the fetus. You can get a sample of the placenta, but this too is not as accurate as going to the source. Amniocentesis will get cells from the fetus by getting amniotic fluid. The best method, though, is percutaneous umbilical cord blood sampling, which will get fetal blood, but it does come at some risk. One day we'll be able to correct genetic diseases with gene therapy. This uses viral vectors to transfer good genes to replace the damaged ones and help these patients. But this is not widely used. It's very experimental. This therapy may also lead to ethical problems, as parents will want to determine most of the genes for their offspring. All right, so now we're going to do some questions. You have already been answering questions in this lecture as you have progressed. Let's look at some together to better show you how to answer questions in this course. Some questions will be straightforward, asking you about the concept directly, but others will try to get you to apply your knowledge and use critical thinking. A 45-year-old Caucasian male has a movement disorder that appears like a dance. It is an autosomal dominant genetic disease he knew he was going to inherit as it runs in his family. What is the diagnosis? What do you think the answer is? The answer is Huntington's disease, also known as Huntington's chorea. If you got it, then good job. Here's another one. An experimental new drug X that has been shown to kill cancer cells works by stopping protein synthesis in those cancerous cells. Which of the following statements about this drug are correct? Notice that this question asks us which statement is correct, and correct is bolded and capitalized to get your attention. What is your answer? D was the correct answer. Remember, ribosomes are responsible for protein synthesis. This was the concept that was being tested. Now for another. A 42-year-old female patient presents with jaundice and elevated liver enzymes. She does not drink alcohol and does not have any known liver disease history. On examination, you see a bluish or dark rings that encircle the iris of the eye. She also has an elevated ceruloplasmin level. What is the likely diagnosis? Again, we have to apply our knowledge here in a lifelike situation. What is your answer? Wilson disease. As I told you before, most of the patients present with liver issues, and this is how the disease is found. The physical exam finding was the Kleisser-Fleischer ring. The ceruloplasmin level elevation was also a characteristic of this disease. Which of the following would not be a symptom or condition in a patient with Marfan Syndrome? What is your answer here? A was the answer. Remember that it was asking which is not a characteristic of Marfan Syndrome. Always read the question carefully. Remembering Marfan Syndrome is also remembering Abraham Lincoln. This ends your first week lecture. Thanks for watching.